Bueno, bienvenidos a todos a este seminario del programa de Frontiers in Genomics, que es organizado por la licenciatura en Ciencias Genómicas, el Centro de Ciencias Genómicas y el Instituto de Biotecnología, todos de la UNAM. Y como saben, pues estos seminarios ahora son híbridos. Las preguntas se pueden formular al final de la presentación, y también las personas que están ya sea en YouTube o por Zoom pueden subir su pregunta en el chat y entonces yo la leeré. ¿Sí? Bueno, para mí es un gusto presentar a la doctora María Colomé-Tatché del Instituto de Biología Computacional del Helmholtz Zentrum en Múnich y del Departamento de Fisiología Química de la Universidad Ludwig Maximilian en Múnich también. La doctora Colomé-Tatché hizo su licenciatura en música en el Conservatorio de Barcelona, en España, y también, interesantemente, hizo una licenciatura y una maestría en física en la Universidad Autónoma de Barcelona. También hizo otra maestría en física en la Escuela Normal Superior y en la Escuela Politécnica de la Universidad de París 6, 7 y 11. Su tesis de doctorado la realizó en un laboratorio de física teórica y trabajó con modelos estadísticos en la Universidad Sud-Orsay de París, en Francia, en donde obtuvo el summa cum laude y se graduó con honores. Después del 2009 al 2010 realizó estudios postdoctorales en el grupo del profesor Santos en Theoretical Quantum Optics en el Instituto de Física Teórica en la Universidad de Leiden, en Hannover, Alemania. Del 2010 al 2013 realizó otro postdoctorado en el grupo del profesor Janssen en el Centro de Bioinformática de la Universidad de Groningen, en Holanda. Del 2013 al 2018 fue investigadora principal y Rosalind Franklin Fellow en el Instituto Europeo para el Estudio de la Biología del Envejecimiento, también en la Universidad de Groningen, en Holanda. A partir del 2016 es Junior Fellow en el School of Life Sciences en la Universidad Técnica de Múnich, en Alemania. También desde el 2016, como lo mencioné al principio, es jefa del grupo de epigenómica computacional en el Instituto de Biología Computacional en el Helmholtz Zentrum en Múnich y profesora de genómica funcional y biología celular en el Biomedical Center de la Facultad de Medicina de la Ludwig Maximilian University en Múnich. A pesar de que es una investigadora muy joven, ha recibido grants y reconocimientos muy importantes, y solo por razones de tiempo voy a mencionar algunos. En 1999 recibió el primer lugar de la 12ª competencia de investigadores jóvenes en España, y como ya lo mencioné anteriormente, también ha recibido el Rosalind Franklin Fellow. Ha tenido varios apoyos muy importantes otorgados por el Helmholtz Center, y uno de ellos es con inteligencia artificial, que me llama mucho la atención. Y también tiene otro en dinámica celular, para mencionar algunos. Ha dado múltiples conferencias por invitación en muchas instituciones y en universidades y congresos. Y el día de hoy nos va a hablar de Single Cell Computational Epigenomics. Y bueno, la plática va a ser en inglés. Y muchas gracias, Marian, por estar aquí con nosotros, y estamos listos para escuchar tu plática.
Hola, ¿funciona? ¿Sí? ¿Me oís? Ok. Muchísimas gracias por la introducción y muchísimas gracias por la invitación. Estoy muy contenta de estar aquí. Es la primera vez que estoy en México y me está gustando muchísimo, con lo que estoy súper feliz de que me hayan invitado. Ayer también lo pasé muy bien hablando con los estudiantes y estoy muy contenta de dar mi charla hoy. Va a ser en inglés porque, aunque yo soy de Barcelona, nunca he dado una charla en español y me da miedo que no podría encontrar las palabras. Entonces, desafortunadamente voy a cambiar ahora al inglés para dar mi charla. Y pues, like it was introduced, I will discuss today about both single cell and single molecule computational epigenomics. So in my group, we are very much interested in the field of single cell epigenomics. And first of all, I will give you like a little bit of a metaphor of why is it that we are interested in working with single cell genomics. So imagine that you wake up every morning and you like to drink a smoothie. And you mix all of your fruits and you drink it every morning, but then there is one day where you suddenly drink it and you think, this smoothie doesn't taste good today. And you cannot really pinpoint what was the fruit in it that was actually really giving you a hard time. If you instead would have prepared a fruit salad, you would be able to actually tell this is the fruit that is actually gone wrong. So this is like the analogy of saying, if we are, for example, studying a disease like cancer, for example, or a developmental system, and we know that something, for example, at some point has gone wrong, we may be interested in knowing which one is actually the cell or the cell type that has caused this disease or has caused this developmental process to go wrong. And if we want to really get to the bottom of it, the best way of doing it is actually to really sequence every single cell at a time and be able to really explore what is happening in every cell type separately. So in general, when we think about single cell, and let's say if I have a colleague of mine that says I've done a single cell experiment, in general, what they mean is that they have done a single cell transcriptomics experiment. So where they have measured the transcriptome, the RNA, the expression of every gene inside of every single cell. I come from the field of epigenomics, and I would tell you, well, if you only sequence the transcriptome, you're basically missing all of the regulatory component that is sitting there in the cell between the genome and the transcriptome, and that will allow you to produce so many different transcriptomes from one and the same genome. And moreover, if you do single cell RNA sequencing, what you are able to measure, it's basically only the expression of the genes, and moreover, in general, only the expression of the most highly expressed genes. So you're missing a lot of information that is happening everywhere else in the genome and that you obviously cannot measure because it's not being expressed. And this is something that you can instead access if you do single cell epigenomics. Wait a minute, there we go. So this is the different, let's say the most popular measurements that one can do at the moment at the single cell level. So as I've mentioned, of course, with the transcriptome, we can measure it with the use of single cell RNA sequencing. We can also measure the genome with the use of single cell DNA sequencing. This is a technique that is being less and less used in the community because basically you're sequencing a lot of DNAs, but of the same organism. So the amount of information that you get, the amount of variation that you get is really very small. In some cases like cancer, it may be interesting, but with the very shallow coverage that we manage to get per single cell, it's still very difficult to really capture the different mutations that may be causing a disease, for example. But it's also possible, single cell DNA-seq. And then here in between the genome and the transcriptome, I have put here the methylome. So it's possible to do the single cell bisulfite sequencing and chromatin architecture. And this is mainly the single cell ATAC-seq. This is the technique which is mainly used at the moment for measuring the openness or the closeness of the chromatin at the single cell level. So as I said, I'm interested in these two layers of information and how they interact with the genome to produce so many different transcriptomes that, let's say, can be seen as a phenotype, a molecular phenotype of the cell. So what I want to tell you about is different things that we can do with the single cell epigenomics data. And the first thing that I will tell you is how we can use single cell epigenomic data to study cell identity. So for doing that, we have developed a package in the group, which is called epiScampi. And I will maybe come to this side. epiScampi stands for epigenomic single cell analysis in Python. This is why it's called pi at the end, epiScampi. And epiScampi can take single cell DNA methylation or single cell ATAC-seq data, and it can also integrate it with single cell RNA-seq data. And we already built it in a very modular fashion in a way that one can then integrate the so-called multi-omics data when you have, for example, the RNA and the ATAC for one and the same experiment. So the first step of our pipeline, it is to do a feature selection, so a genomic feature selection. When people do single cell RNA-seq, they don't even think about feature selection because they have the genes. So basically what they have is that for every cell that they have in their population, they've measured the expression of the genes. This is their feature space, but they don't really think about that. But in single cell transcriptomics, your feature space is the genes. Now, when you start measuring the epigenome, you get information everywhere in the genome, not just at the genes. And then you really need to start asking yourself, how am I going to summarize this data? You cannot go at the single base pair level because that would explode in the computer. So we try to think of different ways that we can summarize all of this genomic data. And what we have done in our program is that we allow a lot of flexibility for the user. And you can, for example, construct features which are windows, which means that you don't want to decide what is in my genome, what am I looking at? No, you just want to take everything. So you partition your genome in, for example, 5,000 base pair windows, and then you summarize how open are these windows or how much DNA methylation level I have in these windows. You can instead decide, no, I actually know a little bit better what's in my system, and I know that I don't want to use the windows, but I want to instead use genomic annotation. And for example, I want to use the DNA methylation level at promoters, because I know that this is going to give me a lot of information on whether the genes are expressed or not, or the DNA methylation at enhancers, or the openness at enhancers, or whether some transcription factor binding sites are open or not. That will give me an idea of what transcription factors are open in that given population, for example. So this would be like an annotation-driven kind of feature space construction. And of course, when we work with single-cell ataxic data, the feature space that we in general always use is the peak feature space. And this means that we take the single-cell ataxic data that we have for every one of the cells separately, we merge them in the computer, and we call peaks like we would do with a classical ataxic experiment. And then for every one of these peaks that we have scored in the bulk sample, then we go into every one of our single cells and we ask, was that peak open or closed in every one of the individual single cells? Yeah, so this is the open chromatin, the peak feature space. Now, once we have decided what feature space we want to use, in our program, we can just type a command, which is called build count matrix. And what it does is that it takes the BAM files or the profiles of every one of the individual single cells, and this kind of BET file, which is the feature space that we are interested in, and it will build the different count matrices. So the count matrices here are every row is a cell, and every column is a feature that we are interested in. Yeah, and the number here, it's going to be the quantification of the epigenome in that single cell for that specific feature. So once we have done this feature selection, what we need to do is a little bit of pre-processing. We, of course, want to get rid of features that are not variable in the population, because these features are not going to explain the variation of all the different cell types. We also want to get rid of cells that have nearly no features which are covered, because these are cells which are probably bad quality, and so on and so forth. So we do a little bit of cleaning here. We get rid of features, we get rid of cells, and what we end up with is a matrix that kind of looks like that. In the columns, we have the different features, and on the rows, we have the different cells, and here the color represents the quantification of the epigenome. Now what we can do is that we can take, these are now vectors. So what we can do is that we can compare every two cells with each other based on these vectors of information, given a given similarity measure, for example, an Euclidean distance or a cosine distance, a correlation-based distance. It doesn't really matter, whatever we think it's most interesting. And then we get a graph that is now cells by cells that tells us how similar are two cells with each other. And now with this graph, we can do a lot of these low dimensional embedding methods, which are based on a graph, such as, for example, the UMAP. So now what we have gone from is a situation where for every cell, I have, let's say, 500,000 peaks, maybe 50,000 peaks after I have cleaned the matrix a little bit, into a low dimensional representation of the data. So every dot here on this UMAP is a single cell. And instead of looking at this single cell in 20,000, 50,000 dimensions, I'm only looking at it in 2 dimensions. So I have embedded this very high dimensional data in a 2-dimensional embedding system. So every dot here is a cell. And of course, what we then want to do is clustering. We want to know what cells are the most similar to each other that I can really decide that they belong to one and the same cluster, one and the same cell type. Yeah, so then we kind of color these plots given the cluster that these cells belong to. And then what we do is some downstream that I will explain to you in more detail with 2 different examples. So the first example that I will tell you about has to do with single-cell DNA methylation data, and the second example is with single-cell ataxic data. Both examples do not come from my lab, because in my lab we do not generate data. We only use data from others. So the first example, it's from an already published paper from the Joe Ecker group, in which they profile more than 3,000 single neurons in the mouse. And just to give you an idea, this data at the single cell level is extremely sparse. We only get information of around 5% of the genome for every single cell in our population. what we do though with that is that we can still get the methylation status of every individual cytosine which is covered in our data set for every one of the single cells. And then we build different count matrices given different feature spaces that we can think about. And then with those count matrices, as I told you, we can go into this low dimensional representation of the data, and then we can do clustering. And we can do that, for example, here on the enhancer feature space, where we check the methylation status of all the different enhancers which have been annotated in the genome. Or we can do it in the promoter feature space, DNA methylation at promoters, DNA methylation at windows, or DNA methylation at gene bodies. So every dot in these plots is a cell, and the same cells are present in these 4 different representations. Yeah, so we just get different embeddings and different clusterings depending on what part of the genome we are interested in. And then we can start comparing them. For example, this is the promoter clustering, and this is the enhancer clustering. And we see that some clusters, for example, cluster here for promoters corresponds exactly to cluster 9 in enhancers. But there are other situations, like for example, this one here, where we see that we have 2 different clusters from the enhancers that have only been called 1 cluster from the promoters. And then we can go back to our embedding, and this would be this cluster here in the promoters that has been perfectly split into 2 separate clusters in the enhancers, for example. Yeah, so this was not our data, we didn't dig any deeper in it, but in principle, depending on the part of the genome that you're looking at, you can get different types of information of what your cells are and how to group them. Not too different, of course, because at the end of the day, it's the same cells. Yeah, what we can also see is that, for example, in this DNA methylation case, the enhancer feature space gave us these clusters which really look very compact. So the cell types were kind of very well defined, while the promoter DNA methylation gave us a little bit more of a continuum of the different cell types. And this can be quantified by using this silhouette score that is higher indeed for the enhancers than for the promoters. So it somehow tells us that in this particular data set, looking at DNA methylation at enhancers gave us a better definition of our cell types. Okay, but so until here, this is a bunch of cells that I colored. And probably if you do an experiment, you will tell me, what are these cells? I don't want colors, I want to know cell types. So to know what cell types we are looking at, what we do is that we need to identify markers. So what we need to do is that we need to rank differentially methylated features between the different clusters. And we can do that on any feature space that we are interested in. We did it in this case with a promoter feature space, just because for us non-biologists, it was easier to then immediately put a promoter with the gene that is next to it and right away kind of get a name for the region that we are looking at. So I will show you an example of it where you have the same cells here, which are also embedded into dimensions. But in these situations, we have not asked for having many clusters, but only having 2 different clusters in the data. So we've split the cells in the largest variation, asking for only 2 different groups of cells. And then we ask what features are the most differentially methylated between these 2 groups of cells. And what we see, for example, already down here is that we have a promoter of the gene NEUROT2, which is differentially methylated between the 2. And we can visualize it in different representations. This is the methylation level here on the y-axis of the orange versus the blue cluster, and we can see that it's much higher. And we can also put the methylation level of every individual cell on top of this 2D representation, such that we can right away see by eye where are the cells with low methylation and where are the cells with high methylation. And then since we know that NEUROT2 is a marker of inhibitory neurons, what we can already conclude is that one of the 2 clusters must be the inhibitory and the other one must be the excitatory neurons. And we can do that deeper and deeper, comparing every small cluster to each other, every small cluster to all the rest, and so on. Every time we get a list of features which are the most differential between the 2. And then with like some knowledge, we can go from this list of genes into deciding which one is the most likely cell type that these cells belong to. So this was this example for DNA methylation. And now I will give you a very short example for the single cell ataxic, so the single cell open chromatin. Again, this is not our data. This is data from the Greenleaf and Chang lab. What they have done is that they have measured the open chromatin of more than 60,000 cells in blood. And again, this data set is extremely sparse because for every one of the single cells, we only have approximately 13,000 unique reads. Yeah, so we have really very little information. What we do here is that we construct the peak count matrix, so the peak accessibility count matrix, and we do like before, that we do a low-dimensional data representation and a clustering. And this is what we obtain here. These are the different cells colored with the different clusters that we found. And the same way as before, we can ask which ones are the peaks which are the most open. Here, this is the openness from 0 to 1 in this blue cell type compared to all the rest. And this would be the peaks which are the most open in this light blue compared to all the rest. And down here, what you see is the peak ID and the gene which is closest to that peak. So the peak ID is the number, and the name here is the gene which is the most close to it. Of course, when we do cell type annotation, we do not only look at 2 peaks, we look at a very long list of peaks, like 50 or 100 peaks. But here for this plot, with 2 peaks, it's more visible, let's say. So then, based on these open peaks, we can decide what cell types belong to every one of the clusters. But the interesting thing is that here, this is, as you can see, you do not really see these very separate cell types as we saw in the brain, because this here is blood, and this is a cell which is in constant differentiation. So actually, there is another way of looking at this data, which is more interesting in this case, which is a thing that we call diffusion pseudotime. Diffusion pseudotime, what it does is that it does like a random walk on your cell population based on the similarity matrix that you have. And it tries to look for the shortest path between any 2 cells. And then it orders them based on the shortest path that you find. And this would be the result here. I know that the colors are not the best, but this is like the beginning. You need to root it somewhere. So this is where we root it. And these are the different branches that go in all of these different dimensions that we call diffusion components. And here I put the same colors that you have here. And what we see is that very nicely, the progenitors are here at the tip. And then in every one of these differentiation branches that the cells have taken, we see that the cells are really ordered all along this branch until we reach the most finally differentiated state for the T cells, B cells, myeloids, and basophils. So this method that I've shown you is what we use to decide what cell types are we looking at when we have single cell epigenomics data. And in our case, we've put it together as a pipeline that we call episcampi that is available in Python. And all of these things that I've been telling you, it's really like one command line at the time. Once you have built this count matrix, it's really like calling differential peaks, doing a differentiation trajectory, and so on. It's really like one command line at a time. And this was published last year in Nature Communications. So until here, I was telling you about how to analyze one data set at a time. But the community is at the moment generating a huge amount of single cell data sets. And on this plot here, that is already old because it finishes in 2017, in red, you can see the data sets that have been generated for single cell ataxic. This is the number of cells. So up here, we are at 1 million cells. And really, data sets with 1 million cells at a time are being generated all the time. And the thing is that there are many different labs in the world that are generating data sets for the same organs or the same tissues or the same diseases. And it's a pity that we cannot take all of them and work with them together because there are too many batch effects between them. So what we decided to do, so there is no method at the moment which has been developed for integrating these single cell ataxic data sets. And what we decided to do was to take methods that have been developed for integrating, for doing batch correction of single cell RNA-seq data and check how well do they perform when we apply them instead to single cell ataxic data. And this is what I will show you now here. So this is about single cell ataxic data integration. So we wanted to make it difficult. We didn't want to just take 2 data sets, but we took 3 of them. Of course, they need to be for the same cell type or the same organism, let's say, because otherwise will be impossible to integrate them. And what we took in that case, we took a data set from the Kuzanovich et al. Paper. Kuzanovich et al. Profiled many different organs and tissues in the mouse, and we took the prefrontal cortex and the cerebellum. Then we took a publicly available data set from the 10x Genomic platform where they had done fresh cortex for adult mouse brain. And then we took another huge data set with nearly 86,000 cells, also from the cortex of 6 different mice. What do we have? We have basically 3 separate count matrices, like I was telling you about. This is the count matrix for the Kusanovich et al. With around 13,000 cells. This will be the 10x count matrix with around 3,000 cells. And this will be the Fang et al. Count matrix, where we only took 50,000 cells to make it a little bit more balanced. And what we need to do is that we basically need to concatenate the matrices to be able to analyze all of the data as if it was coming from one and the same sample. The methods that do that for single-cell RNA-seq data, they just concatenate because their columns are the genes, and the genes are the same for all of the data sets that they are looking at. But here we have chromatin data. So in principle, we need to decide how are we going to concatenate these matrices, because we do not have necessarily the same features in every one of these data sets. So we decided to use 3 different feature spaces. The first one is windows, and that's obviously an easy one because you choose a window size, and this windows is going to be exactly the same one in your 3 data sets. The second one, it's one that was obviously very much driven by our collaborators that were also working on this paper, that were working with single-cell RNA-seq data. And they told us, no, since you're using methods for single-cell RNA-seq, what you should use is what people call gene activity. And gene activity is the quantification of the openness of the promoter and the gene body for every one of the genes, which, as you can imagine, it kind of gives you a good proxy of whether the gene is highly expressed or not. So this is the gene activity. And since we really come from the epigenomic fields, we really wanted to use the peak feature space because we think that this is the best way to describe our data. So what we did in that case, obviously, when you call peaks, they are going to be different in every one of the data sets. So what we did is that we called peaks in every one of the data sets, and then we kind of used some ad hoc method to merge the peaks and decide, what is a new peak in this merged data set. So with these 3 different feature spaces, now we have 3 matrices that we can concatenate. And the first thing that we did is that we asked, how does this 2D representation of my cells look like if I don't do anything? I just take the 3 matrices and I concatenate them with each other. And this is what we get here. The label is very small, but what's important here is that in blue is the 10x genomics, in orange is the Kusanovich, and in green is the Fang et al., like the same colors as used here. And this is the embedding that we get for the window feature space, this for the peak feature space, and this is using this gene activity feature space that you can see really doesn't keep that much information in the data because it's much more diffuse. And what you see is that you can really see like the green blobs and the orange ones and the blue ones are really not integrated on top of each other, but they are just like next to each other. So what we did is that we tested a very long list of methods. It doesn't really matter, you don't need to read it. These are many different methods that people have developed for integrating of single-cell RNA-seq, and we tested on our data set based on a set of metrics that were both testing the batch correction and the biological conservation. So why do we need to test both? Imagine that I have this batch 1 and this batch 2, and in each one of them, I have 3 different cell types, the round ones, the star ones, and the triangle ones. What I could have is a method that does a beautiful integration of my data sets. That would be the method A here. The 2 data sets, the blue and the orange ones, are perfectly mixed, but we have completely forgotten what cell type was what. This is not what we want. This would be a very good batch correction, but would be a terrible biological conservation. What we want is a situation like this method B here, that at the same time conserves the biological information, but also puts the batches on top of each other. Yeah, so this is why we developed many different metrics. You cannot even read it, but like silhouette score, ARI, AMI, homogeneity, and so on, that quantify these different things in your data. And then we quantified every one of these methods based on all of these metrics. Again, I'm not expecting you to read it. These balls here, the bigger they are, the best the methods perform, and the smaller they are, the worse the methods perform. And then we come up with like an aggregated score here that ranked the methods from the best one all the way to the worst one. And just for your information, here is the performance of the unintegrated situation, which tells me that a lot of the methods actually perform worse than just using your data set without integrating it. Yeah, and this is because the biological conservation of the unintegrated data set is obviously very good. So now let's just look at like the top and the bottom ones, just to give you a few examples. So this is the unintegrated one again, and this Liger is our top performing method that has a very good balance between the batch conservation and the biological conservation. And you can see that indeed the two batches are very nicely put on top of each other. These are the three colors here, and here are the cell types. And for example, you can see, which was to us very interesting, this red blob here is these cerebellar granular cells, which were only present in one of the three batches. So this method was very good at integrating the cell types, but also keeping separately and not integrated the cell types that were not present in the other batches, because this is a very common problem when you integrate data, that it really integrates everything. And if you have one cell type which is unique to one batch, it will still integrate it with something else. This is just an example of like a lot of funky results that we got. There are a lot of methods that do not perform well at all. And this here, I just put it as an example because this was the best performing method in the gene feature space. This would be this result here. And to me, this kind of results, what it tells me is that if I'm looking at single cell ataxic data, I should not work with the gene feature space, but I should be looking either at windows or at peaks instead. So this was part of a huge benchmarking that we did where in my group with Chris, who's here, and Anna and me, we were looking at all of this integration of single cell ataxic data. And the group of Fabian Thijs, who's the director of the Institute of Computational Biology, they were checking how good the integration methods do for single cell RNA-seq data, which is what they are actually being developed for. And this was appeared this year at the beginning of the year in Nature Methods. Good, so until here, I've been telling you about the single cell ataxic. And I come really from the epigenetics field. And I'm always a little bit disappointed that nowadays everybody works with single cell ataxic data, because I really come from the DNA methylation field. And I think that you actually lose a lot of information if you only measure openness and closeness. What's really interesting, if you look, for example, at embryonic development, it's really to go and look at the DNA methylation. The problem, though, is that this is extremely easy to perform in the laboratory. It's high throughput, it's droplet based, you do experiments, you just buy a kit, you put your cells in the kit, you get 10,000 cells at the end with the single cell ataxic profiles. If you want to do the single cell bisulfite sequencing, it is plate based, it is extremely expensive, you do your 96 wells every time, your 90 cells with your empty wells and whatnot. So it takes a lot of time, a lot of manpower and a lot of money to develop it. So what we thought in my group is, would there be like an orthogonal way to still access the single cell DNA methylation information without having to do the single cell bisulfite sequencing? And what it occurred to us is that we could use this long read technology, this MinION technology, to actually extract epigenetic clones from the data. So this is why the title of the talk was single cell and single molecule, because MinION is not single cell measurement, but it's single molecule, because you do this super long molecule sequencing of like thousands of base pairs at a time, and you get for free the DNA methylation information on your reads, because it depends, it does some kind of intensity calculation of the read that goes through a pore, and whether the cytosine is methylated or unmethylated, the intensity is different. So you can actually infer the DNA methylation information. So the idea is the following. Imagine that you have a tissue or a tumor, which is actually the idea that we had in mind when we developed this, that has different epigenetic clones in it. That would be the red, the blue, and the lila clone. So you have epiclone A, B, and C. And these lollipops here are the cytosines that are either methylated or unmethylated when it's full or empty. Now, if you really wanted to access the variation, of course, the best would still be to do the single cell bisulfite sequencing. As I said, it's expensive, it's difficult, etc. If you do the typical bisulfite sequencing with Illumina sequencing, what you get is these very short reads. So you would be able to tell me in DMR1, in the region 1, I have, I don't know, 20% methylated, 80% unmethylated. And in DMR2, I have 20% unmethylated and 80% methylated. But you cannot really put them together. You don't know for every single cell, what pattern do I have genome-wide. So the idea is that you have these long nanopore reads. And thanks to these long reads, what you can start to do is to link the methylation information at the left and at the right of the reads. And you know that a single molecule must be coming from a single cell. So because it comes from a single cell, it's single molecule, but you also somehow get single cell information. Yeah, so then if you were able to group them, you would be able to say, oh, these guys come from one clone, another clone, another clone. So what we did in this project is that we kind of hijacked methods that were already developed for doing phasing of polyploid genomes. But what we did is that, so here are my reads. These are these long reads, and on top of it, you do not see it, but there are all the different genetic information, SCGT. What we first do is that we get rid all of the nucleotides, which are not a cytosine. And then what we have is the reads with the information of whether the cytosine is unmethylated or methylated. Then we will, of course, get rid of the positions in the genome that have no variation, because this will not help me to cluster my reads. And we end up with a set of variable CPGs along the genome that have variation in my population. And this is what we then input into an algorithm, which is called nphase, which was developed for phasing of polyploids. It was not developed by us. And this algorithm, what it does is that it scores overlapping reads, in our case, based on their methylation similarity. So it counts how many methylated Cs are the same or not, divided by all the shared variable cytosines in the different regions. So let's say I take two reads, I ask, are they similar enough? And if they are similar enough, they are converted into a consensus sequence that I would call them now AB. And this consensus sequence is then put back into the pot, and it's compared again with all the different reads that overlap with that position, until the situation where I will only find reads which are too far away from the cluster identity, and then they will not be merged anymore. And then at that point, I will stop the merging procedure. What I get as an output here is the number of clusters with the methylation patterns on top of this cluster. So in this toy example here, I would get 3 different clusters, and every one of them has a different consensus methylation profile. Now, to show you how this works, we have applied this method to an in silico mixture of 3 different breast cancer cell lines. So what we do, we have the MinION data, again, publicly available data, MinION data for 3 different breast cancer cell lines. And on the computer, we mix it. And of course, we do not tell the algorithm what cell line was what. And we ask the algorithm, can you separate them? Can you deconvolve them in different clones? Here, I show you an example on chromosome 6. These names here are the 3 different cell lines that we use. And what you can kind of see by eye is that this region here is very red, so highly methylated for the top 2 cell lines, and it's more like blue for the 3rd cell line, while the region here on the right, it's kind of the other way around. So we ask the algorithm, can you split the reads? And this is what we get. We get 3 clones in that region, the blue, the purple, and the orange one. And this is their cluster identity. So what we see is that the orange cluster mainly identifies with one unique cell line, the lila cluster also, and the blue cluster also. So our algorithm had actually really been able to deconvolve these 3 different cell types inside of our mix of cells. There are situations where what we get, this is chromosome 19, we get quite a lot of those, where what we find is that, so we mix all of the reads together, we give them to the computer, and the computer tells us there are only 2 clones in that region. And it turns out that every cell line has exactly 2 clones inside of it. And we are exploring it, thinking that this may be some kind of imprinting, for example, where every cell line has half of the reads that come from the father and are methylated, half that comes from the mother and are unmethylated. So we are exploring how does this correlate with the haplotypes of the different cell lines. So this is work still under construction. We are preparing a manuscript with a PhD student of mine, where we are happy that without having single cell data, we are still able to access all the heterogeneity that is present in samples. And we are also applying it into a mantle cell lymphoma data set from a collaborator of us, where we are trying to look, do we have different clones inside of it, and what regions do this correspond to, etc. Good. So until here, I've been telling you a lot on how to look at different cell types. How can we use single cell data for identifying cell types? But what we were also discussing in the group is all of these single cell epigenomic measurements that we do, they're actually very close to the genome. We measure the epigenome, but at the end what we are doing is that we are sequencing the DNA. With the RNA, transcriptome sequencing, we really only measure the transcripts. If we do single-cell DNA methylation or single-cell ataxic, we actually access the genome every time, different parts of it. So what we thought is that maybe we could exploit that information to call copy number variations in the individual single cells, thanks to the fact that we are every time measuring the genome of the individual single cells. So if you're interested in calling copy number variations, there are many classical measurements like karyotyping, where you get these beautiful plots here, or interface fish. But in general, this is extremely low throughput. You need to have cells that divide, you need to image them in the microscope, and you do not really capture very small copy numbers in your data set. If you want to instead access the whole genome with a lot of resolution, what people have done until now is, for example, doing this array comparative genomic hybridization, ACGH, or exome sequencing, or even whole genome sequencing, where then you count the number of reads and you get this as a proxy of how many copies you have in that region. But of course, you can only do that in bulk. You can only access your population of cells. If you want to know the heterogeneity at the level of copy number variations, you would need to go to single-cell genomic sequencing. And this is obvious, like if you think about it, if you do single-cell DNA sequencing, if you suddenly see that you have a region in the genome that has double amount of reads mapping to it, this tells you that there has been a gain of a chromosome in that region. Or if you have a region that suddenly has half of the amount of reads, this would tell you that there has been a loss in that region. And this is something that we already did in my group a long time ago. In 2016, we had a collaborator that was very much interested in genome architecture and was doing a lot of single-cell DNA sequencing. And we developed back then an algorithm. So this is real data here. This is a few chromosomes, and the peaks here is really the reads mapping at the different locations in the genome. And you could see that indeed there were losses here, there were gains here. You could actually see them by eye. And what we did is that we developed a method that was based on hidden Markov models that was considering different states for the different omis in the genome. And using that model, we could actually tell, this would be a monosomic and this would be a trisomic region. This was based on single-cell DNA methylation, DNA sequencing. Unfortunately, there is less and less people nowadays that do the single-cell DNA sequencing. There used to be a kit also available that you could buy, and you could, again, just buy the kit, put your cells, and get as an output the single-cell DNA sequencing. But this has been discontinued, and I think it's because not a lot of people were actually using it. What people do instead, they do a lot of single-cell RNA-seq and a lot of single-cell ataxic. There have been attempts of calling copy number variations from single-cell RNA-seq data. But as you can imagine, RNA-seq data is very much confounded with the expression of the genes itself. You do not have 2 copies. You can have from 150 copies all the way to 2 copies, let's say. And therefore, it's extremely difficult to infer real copy number variations from the single-cell RNA-seq data. So what we thought is, what about single-cell ataxic data? There is a lot of people that are doing it at the moment. It is genome-based. You measure the peaks, you measure the genome, but it's extremely sparse. But why not? Let's try it. So we took a data set and we decided to start exploring. And through it, we developed an algorithm that we called epi-aneuphinder for looking at aneuploidies in the genome from epigenomic data. And the first thing that the algorithm does is that it counts the reads in bins and it removes blacklisted regions. So we end up with a data set that looks kind of like that. These are all the different chromosomes in the x-axis. And in the y-axis, these are the number of reads that map into different bins in the genome. Then the next step, it is to apply a method which is called binary segmentation using the Anderson-Darling distance to identify breakpoints per chromosome. So this is how it works. This is a piece of toy data that represents these reads that map into the genome. And what we do is that we scan the genome in many different positions. And at every position, we ask, how is the distribution of the data on the left compared to the distribution of the data on the right? And this would be these 2 histograms here. You see they are more or less on top of each other. And you can look at the cumulative distributions here of these 2, the blue and the red distribution, and you can count the distance between them. And the thing is that if you kind of hit that position where the distribution of the reads was the biggest, you can also identify that by having the highest distance between the 2 distributions, left and right. So we do that, we scan the genome many times, every chromosome at a time, and we keep the positions that have the highest Anderson-Darling distance between the left and the right. And then we take the, the mean openness in the genome, and we ask, are these regions higher or lower than this mean openness in the genome? And like that, we cannot really assign copy numbers, whether it's 2 copies, 3 copies, 4 copies, but we can tell it is statistically higher than the mean or lower than the mean. And we put these colors. So to show you that we think that it works, I will show you results from a cell line. This is the SNU cell line. This is again not our data. This has been published by another group. This SNU cell line is known to have a lot of different clones inside of it that have different copy number variations. So we took the data set that had around 3,500 cells, and we did this copy number variation calling. In green here, so sorry, every line is a single cell, every column is a chromosome, and in green means average openness, in blue it means loss, and in red it means gain. So this is what we found here, and we wanted to know how close are we to the reality. And what we did then is that we compare that, it wasn't loading, to a data set from the same cell line that had been profiled using single cell DNA sequencing. This was done in another lab, so not the same lab where we had gotten the first data from, not in the same year. So it's the same cell line, but of course we expect some level of variation. But what you can see is that we basically see the same trends. We see, for example, this big loss in chromosome 3 that has this clonality. Some cells have a longer gain, some cells have a shorter one. We see these 3 bands of loss in chromosome 4. We see some different patterns here. We see some gains that we completely miss in our data set, probably also because ATAC-seq has much less power than DNA sequencing data. We correlated the 2 results. Since these are not the same single cells, what we did is that we built the pseudo-bulk profile for this and for that, and we compared it to each other. And what we found is that they were very highly correlated at nearly 0.9 correlation between the 2. So we were kind of happy, convinced that our algorithm works. It doesn't have as much power as single cell DNA sequencing data, but it seems to be working. And then we went to a real data set, again, not our data set, but a real data set that was not a cell line. And this was a cancer data set from basal cell carcinoma that had been presented in this paper here. And we had in that situation a little bit more than 2,000 cells. And these are these 2,000 cells here with their copy number profile that we identified. What we also did with these cells is that we clustered them based on their similarity of their copy number profiles. And we found groups of cells that seem to have a similar karyotype. For example, a group here that has a loss in chromosome 4 and kind of nothing else, or a group here that seems to be completely disomic, that doesn't have any gain and loss, and so on and so forth. Then what we also did is that we asked for the identity of these cells, like I told you on the first package that we developed in epi-ScanPy. So we embedded these cells here, not based on their copy number profiles, but based on their peaks, like we would do with any data set, single cell data set that we get. And this is the different groups that we found. And by looking at markers, we could identify that the blue ones are tumor cells and the orange ones are fibroblast cells. And then we ask, where are these cells in this karyotype? And we put them here. And you see that the fibroblasts correspond exclusively to one of these clusters, and a little bit here too. And all the other clusters are different types of tumor cells. What we can also do is ask, where are these karyotypes on this 2-dimensional embedding? So now we put this color here, this karyotype color, on top of the embedding. And what we see is a complete mixture of karyotypes. It doesn't look like the karyotype is defining anything about the cell identity when we look at it at the level of the peaks. Yeah, so this tells us that if we do this classical embedding where we take the peaks and the openness for the individual single cells, we actually miss the information of possible different karyotypes that the cells have in the population. And yeah, I mean, this is just a distribution of the different cell types in the different karyotypes. So this is a method that we have developed that it's called epi-ano finder for calling copy number variations in single cell ATAC-seq data. As I tell everyone, if you really have a pure interest in copy number variations, do not do single cell ataxic. This is really not the way to go. But if you have done single cell ataxic for any other purpose, what we give you is an opportunity to yet add another layer of information on your data, which is a proxy for the copy number variations which are present in the different cell types that you have in your data set. So what we show, we have also done some simulations with different coverage, also to see, at what point our algorithm starts to break down and so on. And this is at the moment on bioRxiv, and we actually have it under review at the journal. And with this, I am done. I've presented you different things that you can do if you have generated single cell or single molecule data. And this is, of course, the people that did the work. This is my group. We are split between the university, the LMU, and the Institute of Computational Biology at the ICD. And the people involved are the ones that did most of the work. We did that also in collaboration for the copy number variation with the group where I was before in the Netherlands also. And as I said, for these benchmarking results that I have presented to you, we did that in collaboration with a group of Fabian Thijs, which is also at the Institute of Computational Biology. They took part of all of the single cell RNA-seq part of the benchmarking. And this is the funding and the different graduate schools that I am part of. And thank you very much for your attention. I forgot to put here that I have an open position in my group if someone is interested for a PhD. And we actually have a call from the epigenetics graduate school at the Helmholtz Center, where we have not just me proposing a PhD project, but really many different PIs in the graduate school that propose projects both from the computational, but also from the experimental point of view, only with a focus on epigenomics. And the deadline for the application, I think it's next week. So if you know someone interested in it, I mean, you kind of need to send a CV only, so it's enough time.
Okay, thank you very much for a very nice talk. So now we are open for questions. I think that you can ask either in English or Spanish. Yeah. Okay.
Muchas gracias, la plática estuvo muy interesante. Si quieres te pregunto en inglés para que la respuesta sea más sencilla. My question has to do more with the technical rather than the computational analysis. You talk a lot about ATAC-seq and bisulfite sequencing a little bit, but what about single cell ChIP-seq or CORANTAC? Do you think the technology is still kind of ready for the single cell level, or is it just technically impossible to kind of get there?
Great question. There is a lot of effort in these directions. I think there are two publications that really do the single cell ChIP-seq. ChIP-seq even in bulk, it's quite dirty to do. So at the single cell level, it's really even worse. So I think the first paper that did the single cell ChIP-seq obtained something like 4,000 reads per cell. There was another one that appeared recently that was done by the group of Céline Bayot in Paris that did a much better throughput, so it had many more reads per cell. But now people are trying to actually look at different ways of accessing the histone modification information by, for example, DAM-ID, single cell DAM-ID instead, or there are some new methods which are called cut and tag or cut and run, where you really have like a tag that will identify your modification, will cut the genome there, and then you will sequence where it has been cut, which is similar to the ATAC-seq somehow. And we think that this is more promising, but it's still, maybe some people are doing it already at the single cell level, but so far it was really working well for very small pools of cells, 5, 10 cells. And I know people that are pushing it at the single cell level. I don't know if there are publications on it, but this has been really like a change of mindset to really convince ourselves that probably the ChIP-seq at the single cell level, it's really way too difficult to actually perform. So this is more promising. Yeah.
Muchas gracias.
More questions from the audience? Well, there is one, actually there are a couple of questions in the chat. One is from Eric Cuevas Fernandez. How do you define the enhancers in specific cell types?
In that case that I presented, we didn't do it. We just take a catalog of enhancers. But there are some methods based on single cell data that actually try to define enhancers. And there is one that I find quite nice, which is called Cicero. What you do is that you assume, so you do single cell ataxic, and you assume that you have a lot of variation in your cells. And then you start correlating positions which are open at the same time, even like in trans, in groups of cells. So that this gives you a proxy that, I don't know, I have like this position in trans of a given gene, but I observe that whenever the gene, the promoter is open, that position is also always open. And this you can do if you have like a lot of cells and a lot of variation. And like that, they start to build like this kind of maps with these long range interactions between regions in the genome that they tell you it's a proxy for having an enhancer there. And this was published already a few years ago. It was called Cicero. It's a beautiful package, and you can use it then to try to discover enhancers. But in our case, we just use the catalog of known enhancers in the brain. So easy.
And I suppose that you did the same for the promoters or something similar, because what region you consider to be the promoter? So
yeah,
there is also catalogs for transcription initiation sites, but there are different kinds of promoters, and the distance from the elements required for the function of the promoter are in different positions in reference to the transcription start site.
Exactly.
So then what parameter you use it for?
Exactly.
For define a promoter.
You have a great point. There was a paper that compared briefly this so-called gene activity, and they said that the way that you define your data the best was gene body plus 5,000 base pairs upstream.
Okay.
Which is extremely rough. What we have seen is that depending on the catalog that we use of annotated genes, ensemble, or what is the other one that contains more than ensemble? Now I'm blanking. There is another catalog of human genes that contains many more. You really get very different results, because in one case they've worked a little bit harder on annotating other things. What we are actually doing now at the moment with a small project that we have, we are trying to take ataxic data and look at, for example, long non-coding RNAs or miRNAs or positions in the genome that in general we do not even measure at the level of single cell RNA-seq. And we try to think of it as a proxy. Is it open or closed? Would it be expressed or not? And we are trying to see if this gives us any extra information. This would be, regions in the genome that in general we do not think about and we do not access with a single cell RNA-seq. And we thought, what about going crazy? Even like, for example, taking all of the information which is not in peaks, which we would consider it's complete background, like noise. Is there anything there? Can we still cluster the cells? So we are now playing a little bit with it. And we got a master's student that tried to remap the data using a very well annotated genome, because of course, otherwise you kind of throw away all of the repetitive regions and so on. So we started by first mapping the data a little bit better. And now we are trying to look for different feature spaces and curious to see what comes out of it. Because indeed, the annotations that we use are really rough at the moment.
So there's another question in the chat from Alfonso Barragán. Fabulosa plática, está en español. Fabulosa plática. ¿Encontraste correlación positiva entre transcripción y accesibilidad genómica a nivel de célula única?
¿Otra vez?
Otra vez, lo leímos rápido.
La correlación.
Fabulosa plática. No, ¿encontraste correlación positiva entre transcripción y accesibilidad genómica a nivel de célula única?
Sí, eso también me lo preguntaron ayer los estudiantes. La verdad es que no lo hemos mirado directamente y sería muy interesante mirarlo. La verdad es que tampoco lo hemos hecho porque como siempre miramos datasets que no son nuestros, al final piensas, bueno, ¿qué más da un poco? Pero ahora lo estamos mirando un poco porque hay métodos os he presentado la idea esta de incluir distintos single cell ataxic datasets. Hay métodos que lo que hacen es que te dicen si has medido el single cell RNA-seq y el single cell ataxic para el mismo sistema, pero una vez en un grupo de células y la otra vez en otro grupo de células, pretenden mapear el uno encima del otro para decirte qué células del RNA son las más similares de las células del ATAC. Y lo hacen utilizando únicamente el openness en los promoters. Y a nosotros nos da la impresión de que eso es una forma que realmente no es la mejor manera de correlacionar el ATAC y el RNA. Y estamos ahora comparando diversos métodos que dicen que lo hacen con sistemas de datos por los cuales tenemos el ground truth, con lo que podemos mirar realmente si están uno realmente encima del otro. Los papers en que han mirado más de este tipo de correlación, lo que han resaltado más en el paper es las situaciones en las que no están en agreement el uno con el otro. Por ejemplo, hubo un paper muy interesante hace unos años del grupo de Wolf Reich, que se interesa por embryonic development, y allí lo que mostraron era que el openness a veces predecía la transcripción a nivel temporal y lo llamaban como un poised enhancer, ¿no? La idea de que hay regiones en el genoma que a nivel de transcripción uno no vería el cambio aún, pero a nivel de la cromatina uno ya ve un cambio en la cromatina sin que aún haya habido un cambio en la transcripción. Entonces mi intuición, pero eso es una intuición, es que si uno se mira células que están totalmente diferenciadas, por ejemplo en el cerebro, quizás va a encontrar una correlación muy alta entre el openness y la transcripción. Pero si uno mira, por ejemplo, sistemas que están en desarrollo o en proceso de cambio, creo que ahí la correlación va a ser bastante más baja, porque hay muchas regiones que realmente uno va a preceder al otro, pero no lo hemos mirado directamente.
Una pregunta muy parecida. En relación a eso, en los enhancers, muchos enhancers, la cromatina está abierta, aunque incluso en células donde nunca van a ser activos.
Exacto.
¿Sí? Y igual, por lo tanto, y de hecho a veces hasta están contactando al promotor tridimensionalmente y nunca van a ser activos en ese tipo celular.
Exacto.
Entonces, ahí lo que la gente piensa que se puede usar mejor es la acetilación de la lisina 27 en los enhancers, porque eso ya, y eso sería con ATAC, ¿no?
Exacto.
¿Ok? Para poder hacer single cell.
Exacto, sí, sí. Lo que ahora lo podríamos empezar a investigar a nivel solamente de ATAC es porque desde hace un par de años es posible y es comercialmente available, con lo que no, o sea, a nivel de preparación del protocolo no es muy difícil, hacer en la misma célula única el RNA y el ATAC. Y aquí es cuando, por fin, por así decir, podemos empezar a responder este tipo de preguntas. El data es tan sparse que es realmente muy difícil a nivel de una célula, porque si uno, el ATAC es como la RNA, si uno tiene un 0, no sabe si es cerrado o si es missing data. Pero si uno tiene suficientemente células, puedes empezar a hacer estadística y empezar a preguntar este tipo de cosas.
¿Más preguntas? Bueno, me parece ya no hay, tampoco
Perfecto.
en el chat. Muchas gracias por tan bonita plática.
Gracias por la invitación.
Gracias a todos por estar aquí.
Gracias.